Good morning. Thanks very much, Ilia, for the kind invitation. And I'm very sorry I cannot attend in presence this so important and interesting meeting. And I like to present the video of my presentation of title Flavonols from Blueberries and the Grapes Effect on Brain Function. So thank you very much again, and I wish all of you a great success for the outstanding outcome of this important meeting. I like to shed light on my journal. I'm editor-in-chief of Open Medicine. I would be very happy to have some your contribution. Please don't hesitate. And also I'm in the editorial board of important journal like Aging Research Review, Current Neuropharmacology, Free Radical Biology Medicine, and in all my research history, I have dedicated my best attention to the role of nutrients on brain health and disease. In particular, aging, which is at the frontier of science. And these are my last book, being the president of the European Society of Anti-Aging Medicine. We have dedicated a lot of major interest to longevity mechanism in order to assure that life expectancy expanding, not only adding a year to life, but also a life to year. This is one recent book of couple of months ago, the roadmap to longevity medicine, but also 3 weeks ago, brain and mental health in aging. Where we have dedicated our contribution focusing on phytochemicals and vitagens for healthy brain, together with Naomi and my great collaborators that I thank very much. Also, in the last year, we have published books with Suresh Ratan, friend and collaborator, explaining health across science, nutrition, food and diet in aging and longevity, and also human aging. So there is a rationale leading my research, connecting anti-aging medicine strategies from bench to clinics with antioxidants and oxidant disorders. And in particular today, I will present data regarding the nutritional use of memophenol, which is a standardized polyphenol-rich extract from grapes and blueberries in a model of rat model of Alzheimer's disease. So in our study, we have developed with aluminum chloride for 60 days, and then the last months we have treated with memophenol, and then we have evaluated all behavioral and biochemical markers of oxidative stress and Alzheimer's disease neuropathology. As we can see, there is a very significant amelioration of behavioral deficit that these animals develops. Here we can see the latency at the escape time, which is significantly reduced after memophenol treatment. Together with the measure behavioral finding, we can see how neuronal loss is significant in the model, but significantly reduced after memophenol supplementation. Here we can see sham, where the neuronal population are not affected by memophenol treatment. And also, we have measured APP and phosphorylated tau in the hippocampus. And here we can see how in the animals there's a significant accumulation of both APP and phosphorylated tau, and the significant protection afforded by memophenol treatment. Together with a beta deposition, we have looked at NRF2 expression and NRF2 dependent beta genes, including hemoxygenase. And here we can see how in AD there's a response, cellular stress response mobilization, but it's not efficient, while memophenol treatment bars the NRF2 translocation, so it's consistent with activation of the expression of hemoxygenase 1, which is an NRF2 dependent protein. And together with hemoxygenase, we have measured decreased activity of major antioxidant enzymes and the protection by memophenol. Here we can see that this is the case for superoxide dismutase, but also catalase, and we have also measured the glutathione peroxidase, and all antioxidant response is significantly ameliorated by memophenol treatment. Here we can see that together with the measure redox neuronal redox change observed after aluminum chloride and added pathology, neuropathology development, and the significant protection observed after memophenol treatment. We have also investigated the astroglial reactivity and microglial reactivity. As we can see, GFAP increases with aluminum chloride, and memophenol induces significant protection, together with the AKB, which is the protein associated with NFKB that follow the same trend, increase and protection. But also measure pro-inflammatory cytokines such as TNF alpha, interleukin beta, and interleukin 6. So this is a very important observation, which I take the opportunity to thank Professor Osakabe, because we have developed in a laboratory the effect of memophenol on flow-mediated vasodilation. And here we can see how before treatment respect to which administration of polyphenols from berry and grape induce a significant consistent increase in blood perfusion in this model of muscle cremaster in blood irrigation, which is well characterized in the laboratory of Professor Osakabe. Together with this observation, we have expanded our interest on the use of hormetic nutrients in brain health and disease, and we have recently demonstrated the efficacy of boswellic acid from frankincense from Oman, from Boswellia sagra, in regulate autophagy and neuroprotection in a model of traumatic brain injury. This is the paper recently published, and the group with which I enjoy collaborating. So the idea that I'm moving forward with my research is that we have increase in life expectancy, but only increase in the amount of years, just because we have removed the passive causes of cell death, of death, and like antibiotic, like better environmental condition, like better nutrition. We have obtained this shift from 50 years life expectancy at beginning of our century. Today is more than 86 for women and 84 for men. This is only the effect of removal of passive causes of death. Now we want to add not only year to life, but life to year, the quality of life in centenarian, for instance, because I can have two centenarian in front of me, one well being state, another completely in full disease and with risk of death within days. Both are model of successful aging, but yet they express a completely different phenotype. So we want to impact the quality of life during aging in order to obtain not only expansion of amount of years, but also the quality of life. We can do this if we have a good target that we can modulate with proper intervention. One idea is the idea of Vita genes that are NRF2 dependent proteins, and the ability that we have to modulate in a redox sensitive way through NRF2. And this is the case for sulforaphane that modulate the redox state of KEAP1, thereby activating NRF2. In fact, NRF2 freed moving to the nucleus, where binding to antioxidant responsive element, which is in the promoter of critical stress protective gene, which I have defined Vita gene. So these are my Vita genes, include the hemoxygenase 1, heat shock protein 72, gamma glutamyl lyase, which is a critical enzyme in the Meister cycle of glutathione synthesis, but also thioredoxin reductase and sirtuin. So and as a general approach, the biochemical way how measure all polyphenols act is according to the principle of hormesis, where at low dose we have a beneficial effect which we don't see at higher dose, in the higher dose range where we observe a detrimental effect. And this is the way our mental performance works, our lifespan works, and this is the reason why we are so interested in better and further refinement of the idea of hormesis, underlying the measure nonlinear biological response from brain health to brain disease to lifespan. And the way how we can do this is with a nutritional approach, where the dose response of hormesis reflect the beneficial effect of low dose stimulation by hormetic nutrients, where at higher dose we have toxic effect. This is the case, for instance, for memory enhancing drugs, where we can define at low level dose range the beneficial window of their effect, followed at higher dose by toxic effect. Hormesis also has been recognized as the key to conjugate the traditional Chinese medicine, the Eastern medicine with modern Western medicine. And the key unifying the beneficial of medical perspectives and approach is hormesis. Cellular stress response, the hormesis paradigm, and Vita genes are central to my enthusiasm in laboratory research. and we have characterized a lot of flavonoids, nutritional hormetic nutrients according to the general principle of hormetic response. Also, gasobiology, neurogasobiology comply with the principle of hormesis. At low dose, nitric oxide is beneficial, but higher dose it's toxic. But also carbon monoxide from hemoxygenase and hydrogen sulfide from enzyme that we have in our body that generates hydrogen sulfide. So cystathionine beta lyase, cystathionine beta ligase are a protein example of gene expression connected with production of a low dose amount of gas that accomplish important function in especially in brain. We have also used in our experimental approach from bench to clinic also polyphenols present in mushrooms, and we have been the first to document the presence of polyphenols in mushrooms, and we have used it in the clinical trial, for instance, in Meniere disease, which we have developed as a model of neurodegenerative damage, and we have observed the important neuroprotective effect after 3 months of treatment with mushroom that are vehicle of polyphenols. As the last part of my talk concern what is the measure focused in the research in my laboratory, we use lipidomics, we use metabolomics to characterize in the patients, in healthy subject, the pro-inflammatory, anti-inflammatory status, and so we can characterize in real time, day by day, the redox stress response with urinary analysis of any individuals. So we have applied this in autism, where we have performed studies employing antioxidants, and in particular, this is the last minute of my talk, we are developing biotechnological mini organ technology, and in this case we have published thanks to the collaboration with Thomas Hartung, Sergio Modafferi at Johns Hopkins University in Baltimore, and in my lab we are able to develop a mini brain towards a three-dimensional modeling of human brain in health and disease. In particular, these are my mini brain, where we can study all the brain functions, just taking from urine from peripheral blood cell and reprogramming. And after cellular reprogramming, we obtain the brain of this subject. Not only, but the future is based according to the principle of bank power. We can use this brain-rich in stem cells to rejuvenate the own individual, because this brain can secrete microRNA, extracellular vesicle, and a lot of other magic components that will make our future bright. Thank you so much for your attention.
Thanks.